Thank you very much. So I might not get through all my slides because I want to leave time for questions and answers, right? So I'd like to start by thanking the Lawson family and their foundation for their generous support. I hope the series will, you'll share some of the enthusiasm of the work that's going on at the various centers around the University of Toronto. So the interesting thing as I sat and listened to Richard is, you know, students present data, supervisors kind of comment on data. So it'd be a different tone of talk. I do work in the area of probiotics and prebiotics, and I do have conflicts. We work in the discovery research that you saw for Richard and taking it to implementing in humans, children, and adults. So part of the translational, that's the fun part about working at a children's hospital, is to taking it to the bedside. So the research, this is for everyone, but especially for the Lawson family, the research that's going on now isn't pie-in-the-sky stuff. It's stuff that's really impacting on human subjects now. And I think that's really the message of what my talk is. I'm also promised, Richard commented to you on oligosaccharides that microbes can use, or they work directly to improve health. What I'm going to talk about is using microbes to improve health. So those are two, not separate, but interrelated and distinct topics that are very of interest. So the first thing about the microbiome, because this is a huge area, there were 2,000 papers published last year, and the historical perspective I think is really cool, because I don't know how many of you actually know this. The microbiome was first, there's 7 billion people, plus or minus, on planet Earth. And the first 3 people on planet Earth that were sequenced, they're not the human genes, but the bacterial genes, was from poop samples of Canadians. Did you know that? So mostly, yeah, yay. And not only that, they are from Manitoba. They're prairie Canadians. I had written in previous slides they're from Winnipeg, but when I talked to Charles Bernstein, who did these studies, they were coded, so it wasn't actually clear where. They're from Manitoba, we know that. And this first study on 3 people has actually been verified in thousands of subjects and probably $1 billion worth later. But these 3 people, published in Science, describe what's mostly known about humans. 500 to 1,000 bacteria, all 3 Canadians, but some, about half of human subjects have archaeobacteria. There are lots of viruses, prophages, and phages, and fungi present in a microbiome. It's complicated. It's a very complicated space. Many of these bacteria were not described in the pre-genomic era because they were hard to culture. They're not impossible to culture, but they're hard to culture. And there's a lot of distinctions. The second study that was published showed that there was a microbiome, 500 to 1,000 bacteria, but very different composition. So the one thing we know about microbes is they differ from where you are in the world, how old you are, whether you're urban or rural. Richard mentioned whether you're born vaginally or by C-section, whether you're breast, what you eat impacts. And the other big factor is antibiotics. Antibiotics are a huge impact on the kind of microbes you have and how diverse a microbiome you have. So these are the studies that are underway, have been published in really high-impact journals from around the world. The Canadian contribution isn't minuscule, but it's not the biggest contribution either. Lots of work has been published and lots of criticism about the work published, because up to now, it's been a cataloging exercise. A lot of money describing who's there. And there's a huge debate on whether that's a good thing or not. But the current state of the art, I think, my own view, that's the commentary part instead of showing data. And what I think the center is looking at is not just who's there, but what are they doing? What's their function in health and in various disease states? So I think that's the current concept of what the Child Center is looking at, and the microbiome in particular. So to summarize a whole complicated literature, I think the one thing to say is that reduced bacterial diversity, the less bacteria present, is associated with an unhealthy gut and a whole variety of disease states. Now, these studies in humans are mostly correlative. So they're not cause and effect, they're only associations. So they don't prove that the altered microbiome caused the disease. It could be very much the other way around. The altered microbiome is a result of the disease. So animals, complementary animal studies, like the kind Richard showed, are pointing the direction that, in fact, it is causal, that the altered microbiome precedes and results in this case, the animal features that are consistent with human disease, including diabetes, obesity, variety of cancers, inflammatory bowel disease, necrotizing enterocolitis, and I'm going to show you some. I can't cover all this, right? So I have to pick something to talk about. And I thought diet is too close to nutritional sciences. Somebody might know something in the audience and criticize me. So I thought I would go with an area you guys might not know that much about, and it's a really hot area. But you actually do know it, but you might not have thought of it in this way. And that is that there's a brain-gut communication. You already know that because you know when you get up to give a talk and you get a little anxious, you get an upset stomach and you have diarrhea, that's your brain communicating with your gut. But it's also really clear, and I'll show you some data if I have time, that it works the other way around. The gut and the microbiome sitting in the lumen of the gut communicates with the brain. Okay, and that's really what I'm going to present to you today. It's a two-way bi-directional communication, and impacts here impact the gut, and impacts here affect the brain. That's an amazing kind of news story to think about. So this is my, for the nutritional science folks in the audience, diet has a huge impact on a microbiome of a mouse, and it does of a human too. But it's not in isolation, it's very much related, it's not either or. The genome that you have, the genes that you have, are very impacted. Now, the one thing to say about diet is the diet you have early in life impacts your microbiome for the rest of your life. So if you're born in Bangladesh and you come to Canada, even though you're eating Canadian food, you have a Bangladeshi microbiome. Or you can identify where you came from. And vice versa. If you're, and this is a study done a long time ago before genetics. If you take a New Yorker who's been living in Bangladesh for 30 years, they have a New York microbiome. So it's kind of cool. So you can make minor adjustments with the diet, like high fat, but the major impact is decided early in life. So that's why child health is so important, because the developing organism has a huge impact on the immune system and, for the rest of their life. The impacts on adult health and disease are decided early in life. Now, the disclosure that I didn't have on my slide is I'm a pediatrician, right? So I'm a little bit biased in how I view the world. So the studies about the brain-gut connection that I'm going to show you, I've been involved in some of these. There's lots of people around the world. But the lady who did this work in my lab is shown here, Melanie Garreau, and I'm very proud of her because she just got in the last month her R01 grant, which is the NIH equivalent of a CIHR grant. She has a faculty appointment at UC Davis, and she's doing great work. Her studies with me really related to looking at taking a germ-free mouse and see if the altered microbiome affected their brain. And the short answer to the details of the study is it does. They don't have the same kind of response that a same strain of a mouse that's got a microbiome. They don't behave the same. They're more shy. They're more anxious. And their brain function, if you look at a variety of factors, but just showing one example here, is different in a germ-free mouse as compared to a colonized mouse. So the microbiome, the presence of a microbiome, no matter how diverse it is, but the presence or absence has a huge impact distally up here in the central nervous system. So those were my study. Is it true in a human? So we don't have all the answers, but there's a bunch of studies shown here as data in adults, but it's been shown in children and in infants, that the microbiome composition, the ones in gray are different than the ones in black. The less diverse, it turns out, off to the left. And those are the adult subjects with irritable bowel syndrome. So they have unexplained abdominal pain, and we think it relates in some way to their brain. Now, how could that be? Well, this is a subject of a lot of debate. So this is a current review. Reviews like this with lots of complex hypotheses means we don't really know precisely how they work. But the current thinking is that it could involve the immune system, because we know that there is an immune system in the brain now. It could be, despite a blood-brain barrier, the circulatory system. But the most interesting thing, and these are Canadian studies done by John Bienenstock, who's in the Canadian Medical Hall of Fame and might be a speaker one day in this session, is if you cut the vagus nerve, now in mice, not humans, you block, well, you can do a vagotomy in a human too, so you could do these studies. But his studies weren't in mice. If you cut the vagus nerve in a mice, the impact of the microbiome on the brain disappears. So there is the possibility of at least complicated space, but neural, immune, and circulatory hormonal effects on the gut-brain axis. This has become such a change in thinking that this is gastroenterology, the premier GI textbook, journal that's out there. And disorders like irritable bowel syndrome are called functional gastrointestinal disorders. So they're not necessarily diseases. They're symptoms of unknown etiology. They meet in Rome over decades, and they meet multiple times. So there's been 1, 2, 3. And the interesting thing about Rome 4 is they proposed a change in the name. So it's just come out, and I don't know whether this will catch fire or not, but they're going to change the name of these group of disorders like irritable bowel syndrome from functional GI disorders to disorders of gut-brain interaction. Kind of amazing, right? So I don't know what the acronym is going to be for that, but people are now accepting there is a connection. So if there is a connection, how can one alter the microbiome? Can one alter the microbiome? So you already know the answer is yes, because Richard showed you that prebiotics can alter the microbiome. There are lots of people who are interested on the far left in using fecal transplants, which is taking your wife or your husband's, your significant other's microbiome and putting it into you. Does that work? I'll show you evidence that in certain settings, it amazingly works. Is it safe? Different story. We don't know that, but does it work? Yes. Can you use consortium of bacteria or single bacteria to change the microbiome? This is the idea of probiotics, live beneficial microbes that you take and transiently colonize and alter the composition of the microbiome and the function. That's what I'm going to show you. It's not just compositional change, it's functional change. And is it possible to use a bioactive compound from not a live organism anymore, but the active ingredient of a microbe? This one at the top there is said to be an anti-inflammatory compound, and it's been patented by a university, Vanderbilt. So you can't patent the bugs necessarily, but if you find a bioactive compound, definitely possibilities for commercialization. So Richard told you about prebiotics and human milk oligosaccharides. Well, this is already in the marketplace. So if you go to the pharmacy, or if you have your own baby and you look at infant formula, there's now infant formula that have FOS on them, fructooligosaccharides. That's the prebiotics that Richard was talking about. And this is not available in Canada, but it's amazing. It's already marketed in America with this thing HMO on it. So I would say the evidence that you saw Richard present doesn't support that you really should stick HMO on here, because really what this is one little compound taken out of that complex mixture that Richard described to you. So is it enough to call it an HMO? I don't really think it is, but it's being marketed in America like this. So that was prebiotics. Fecal microbial transplantation, it's already being used in the setting of recurrent infection. It's a Clostridium difficile infection. The elderly really suffer with this infection. And it shows that if you give a donor transplant, you can have a huge impact on quality of life. You change the microbiome, and you change the diarrheal disease that people have. The interesting thing is, what microbiome are you supposed to use as the donor microbiome? We thought it was supposed to be healthy people, and who was the healthy person, and what constitutes a healthy microbiome. But it turns out this study is kind of controversial, because it shows that not in one center, but in another center, an autologous transplant does just as well. So what's the bioactive compound? How does it really work? We don't know. It's the observation without the mechanisms really behind it. So transplantation in the setting of inflammatory bowel disease, 2 studies back-to-back in this journal of gastroenterology, one from Canada saying it works, one from Europe saying it doesn't work. So We don't know what the final verdict, we don't know what the final verdict is, but the study that's going on right now, funded by CIHR, Canadian taxpayers, is called the Spore Imagine Network. And this network is looking at brain-gut interaction and the impact of fecal transplants on the brain-gut interaction in IBD, irritable bowel syndrome, C. difficile. So it's a mixture of gastroenterologists, pediatricians, and the lady on the top right, a psychiatrist who knows something about the gut. So we'll know the answers better on does it really work and how does it work coming shortly. But for the nutrition crowd, I thought I would show one study, human study. There are lots of animal. But this human study is really interesting because what it shows is you can change on the y-axis insulin sensitivity with a fecal transplant from a healthy donor. Whereas in this case, the autologous, your own feces as a transplant into the gut had no effect on insulin sensitivity. So it's very provocative, interesting study. The problem with it is it's only 9 people, and it's only in one center, and it's only for 6 weeks. And it's not that long, I guess, 4 or 5 years, but it's never been replicated. So nobody else has shown this yet. And so somebody's got to look at this and see, is it true or is it a one-off? So very quickly, can probiotics be used to alter? These are beneficial microbes. And I think I'm going to skip a lot of these slides to show you that we are doing based on laboratory studies in vitro, like Richard showed you, and in vivo in animal models, we now have CIHR funding to look in human Canadian children to see whether probiotics will have an impact on disease. So I can't show you the results. It's interesting that papers now, you publish the methods first, then you do the study, and then the results come out after. So that's a methods paper of how we're going to do this study, but no results. This is showing, if you do a meta-analysis of all the studies that have been done in probiotics in the setting of irritable bowel syndrome now, it's very effective. The summary odds ratio at the bottom does not cross 1, which means it's significant it is statistically significant, and it's clinically significant because you only need 7. Treat 7 patients and 1 patient will get better. In clinical medicine, that's a pretty good track record. This study was an Irish study of women with irritable bowel syndrome. It shows a dose-response effect. And this study, not the meta-analysis on many more patients, was enough for Health Canada to actually approve probiotics. So Canada is the first country in the world that I know of that's actually approved probiotics for use in a functional GI disorder, or this brain-gut disorder. And Health Canada approved these 2 products. And I can show you the brand names because nobody remembers the bacterial name because it's been approved for CME. And the interesting thing is the bacterial name there on the bottom one is actually misnamed. So when the bug was sequenced, which was after Health Canada gave the approval, it's actually not B. infantis. So it turns out that it's going to have to be renamed and the slide updated. So these bugs work in children and in infants, and how they work, the mechanism of action is probably through local effect on pain channels. So the culture supernatants of these bugs affect pain channels. They have something to do early in life because you can impact the microbiome of baby mice by changing the microbiome of their mothers. And the issue is, so that's the functional GI disorders like colic and irritable bowel syndrome. The big controversy right now, which you might have read about it because it's in the paper all the time, is what about primary brain disorders like depression or autism in children? And here, I think there's a lot less evidence. This is, I would say, very preliminary evidence that the microbiome is altered. The studies in mice were done a number of years ago. These reviews in autism in humans, actually, there's no data in humans. They only cite the mouse studies. So it kind of bothers me a little bit to not see this have extended. Because for parents of children with autism, this is a huge deal because there is no current therapy. They're desperate to use something. But my own feeling is that the evidence is not yet compelling. There's mouse interesting studies suggesting the microbiome might be altered, and giving something like prebiotics and probiotics might be beneficial. But we're a long way from changing clinical practice. So the pipeline for using microbiome-derived treatments like probiotics, I think, follows this good suggestion that you should use in vitro laboratory studies in preclinical trials in the appropriate animal models before jumping straight into human studies. If you want to know about what probiotic to use in Canada, I'm not pushing one that's right or wrong. There's a website that tells you what's out there. And so the take-home messages are, we think there's more going on about the gut-brain interaction, and the diet might have something to do with it. It probably has impact if the microbiome is altered and reduced in diversity in human disease, including brain diseases. That prebiotics that Richard talked about and probiotics, beneficial microbes that I mentioned quickly, might be beneficial, but we need to compare various strains, prebiotics against probiotics, the combination of the two, and we need to be sure they're safe. Like, are these really safe, especially live organisms, and especially early in life when you're impacting on a developing immune system? So the best summary, if you really want a great summary, David Suzuki has done a Nature of Things on this, and I highly recommend it. It's a good take on this. And the Canadian Pediatric Society, what do physicians have to say? We need more research. So that's where the Lawson Foundation and University of Toronto are arm in arm, because we need to advance understanding to impact on application. And with that, I'll conclude, and thank you very much for your attention, and I hope I left some time for questions.